Malaria. The word itself conjures images of fever, chills, and widespread illness. But what exactly causes this formidable disease that continues to impact millions globally, especially here in Ghana? It's not a virus, nor is it a bacterium. The culprit is a fascinating and complex organism known as the Plasmodium parasite. More than just a bug, the protist puzzle. Imagine a single-celled organism that thrives by demanding hospitality. That's the Plasmodium parasite for you. Unlike viruses or bacteria, these parasites belong to a unique kingdom of organisms called protists, specifically protozoa. They are animal-like, single-celled entities, yet, surprisingly, they possess many features reminiscent of plants, with deep connections to algae. This unexpected lineage hints at a complex evolutionary journey, providing clues to their unique metabolic pathways. The ultimate scavengers, a life of dependency. One of the most defining characteristics of Plasmodium is its absolute reliance on a host. There's no free-living form of the malaria parasite. They are always nestled within a host cell. Over time, they've evolved to become expert scavengers, losing the ability to synthesize certain vital nutrients because they've had constant, easy access to them from various hosts. This dependency, however, also presents potential vulnerabilities we can exploit. The mosquito connection, an unbreakable bond. You simply cannot discuss malaria without acknowledging its essential partner in crime, the mosquito. These insects are the vectors, the indispensable couriers that transmit malaria from one host to another. Understanding this intricate relationship is crucial for breaking the transmission cycle. A family of foes, the Plasmodium diversity. The complexity of malaria lies not just in the parasite's nature, but also in its diversity. The disease is caused by over 100 different species within the Plasmodium genus, each tending to be highly host-specific. So, for example, some infect only birds, while others rodents or even lizards. However, a select few have mastered the art of infecting humans. For this discussion, we'll focus on the most notorious of them all, Plasmodium falciparum. This species is responsible for the majority of malaria-related deaths and is a critical focus for researchers globally, not least because it can be successfully grown and studied in the lab. Shout out to Janssen for establishing the first lab-adaptable Plasmodium parasite that we all now use. This ability to culture Plasmodium falciparum has been a game changer, allowing scientists to genetically modify these parasites, label proteins and metabolites, and unravel the secrets of their biology. Beyond P. falciparum, other human infecting species include Plasmodium ovale, Plasmodium malariae, and the increasingly concerning Plasmodium knowlesi. What's particularly sobering about P. knowlesi is its recent jump into human populations. Just a decade ago or a few decades ago, it was exclusively known to infect non-human primates, particularly monkeys. This serves as a stark reminder, much like the emergence of certain viruses, that other non-human primate malaria species always have the potential to cross into the human population. The parasite's grand tour, a 3-stage life cycle. To truly grasp the impact of malaria, we need to understand the parasite's intricate life cycle. It's a journey that can be broken down into 3 main stages. The mosquito stage. The cycle begins when an infected mosquito bites a human, injecting a surprisingly small number of parasites, tens to hundreds of sporozoites, that's the form of the parasite that the mosquito gives you, into the skin. The second stage, the human liver stage. Once injected, only a minority of these parasites actually make it to the liver. Here in the liver cells, the parasites undergo an explosive round of replication, multiplying into thousands of new parasites called merozoites. Crucially, this liver stage is asymptomatic. You won't feel sick just yet. Third stage, the human blood stage. These merozoites, once released from the liver, are primed to infect red blood cells. In the case of P. falciparum, this blood stage cycle is incredibly rapid, taking approximately 48 hours. Within this time, the parasite invades a red blood cell, uses its nutrients to replicate and divide, and then bursts forth, releasing about 12 to 24 new progeny. This cycle repeats, leading to an exponential explosion in parasite numbers from millions to billions and even trillions. This is when people get sick, understandably. The classic hallmarks of malaria, such as periodic fevers, anemia, and an enlarged spleen, are all manifestations of this blood stage infection. So how do we deal with this parasite? Well, we use a multi-pronged approach to control it. Stopping malaria is a complex challenge that requires a holistic view, targeting the parasite, the mosquito, and the human host. Interventions currently in place include vector control. These are measures like insecticide-treated bed nets and residual spraying that are crucial for controlling mosquito populations. Therapeutics. While medicines exist, developing effective treatment is complicated by several factors. Species diversity. Different Plasmodium species have distinct biologies and metabolisms. A drug effective against P. falciparum might not work for P. vivax. Cellular preferences. Some species infect older red blood cells, while others target only immature red blood cells called reticulocytes. Life cycle stages. The metabolic needs of the parasite can vary significantly between the liver and blood stages. A metabolic mosaic, the Plasmodium's inner workings. So adding to the complexity, the Plasmodium parasite is a metabolic mosaic, possessing several distinct compartments, each contributing to its intricate metabolism. Decades ago, researchers made a groundbreaking discovery. Plasmodium has 3 different genomes, 3 distinct sources of genetic material. 1, a classic nuclear genome containing about 5,000 genes. 2, a mitochondrial genome, similar to our own cellular powerhouses. But thirdly, they have the apicoplast or plastid. This is perhaps the most unexpected and fascinating discovery. The organelle is surrounded by 4 membranes and contains its own genetic material and metabolic pathways. It harkens back to the plant world and ultimately to the bacterial world, revealing the parasite's unique evolutionary adaptations. This plant-like organelle is a significant source of metabolic pathways crucial for the parasite's survival, making it a key target for drug development. So let's leave it here for this video. If you enjoyed it, you may want to check out the Plasmodium life cycle that's already been covered, and stay tuned for the next phase where we talk about resistance genes in the parasite. Catch you in the next one. God bless.
Be strong.